Hello everyone. Here today we will discuss on molecular techniques, various university questions on molecular techniques. First techniques, let's see polymerase chain reaction, PCR. So the question would be like, mention the principle and application of PCR. So first you need to give the definition for PCR, that is the polymerase chain reaction. It is an in vitro DNA amplification procedure. Millions of copies of particular sequence of DNA can be produced within a few hours. So from a single DNA we can produce its million copies. Millions of copies of that DNA can be produced within a few hours using this techniques. Let's see what is the principle and the procedure behind this. So you can explain the steps. Step 1 is denaturation steps or separation. DNA strand is separated and it is melted by heating at 95 degrees Celsius for 15 seconds to 2 minutes. Then next step is annealing or priming. The primer are annealed by cooling at 50 degrees Celsius for half minute to 2 minutes. The primer hybridized with their complementary single stranded DNA produced in the first step. So first we are separating and melting the DNA strands. Then we are annealing it with the primers. Primers will hybridize with the complementary strands of the DNA. Next step we are polymerizing. Third step, polymerization. New strands, DNA strands are synthesized by Taq polymerase. And next step, that is the repetition of step 1, 2 and 3. So by each steps the DNA strand is doubled. Next step, that is the fifth step, that is after the amplification procedure, DNA hybridization techniques or southern blot technique is used, and it is an southern blot analysis with a suitable probe is done, and it shows the presence of DNA in the sample tissue. Next we can explain about the applications. Applications of PCR. First one is diagnosis of bacterial and viral diseases. Bacterial infections, whether if it is in the small bacteria, if it is in the small quantity or larger quantity, we can identify the presence of those bacteria by using PCR technique. For example, in tuberculosis, in the early stage, the sputum will be containing only very few bacilli. So the acid fast staining test may be negative because of the low quantity of bacteria. But if we are doing PCR, if we are amplifying the DNA using the PCR technique, we can give a positive result for the early phase of tuberculosis. And various other, not only tuberculosis, various other bacterial infections can also be detected. Here we are separating the specific nucleotide sequence, and it is of that bacterial infection causing bacteria, that is bacilli, is amplified by PCR, and then it is detected by using southern blot technique. Next application, it is in the medico-legal cases, that is during crime scenes. DNA from a single strand or even from a hair follicles can be analyzed. The restriction analysis of DNA from a hair follicles from the crime scene is studied after PCR amplification. This pattern is then compared with the restriction analysis of DNA sample which is obtained from various other suspects. The culprit sample will preferably match with that of PCR amplified sample. So even from a blood drop or a blood stain or dead skin or from a hair follicle, the DNA can be separated and can be amplified and can be compared with that of the suspects. Next application, in genetic disorders, diagnosis of genetic disorders. This technique is used to amplify gene segments that contains known mutations for the diagnosis of various inherited diseases such as sickle cell anemia, thalassemia, cystic fibrosis, etc. Next is in prenatal diagnosis, in prenatal studies of inherited diseases. Amniotic fluids analysis uses PCR technique. Then next one, in cancer detection, PCR is used to monitor residual abnormal cells present in treated patients. Similarly, identification of mutations in oncosuppressor genes such as P53 or retinoblastoma genes can help to identify the individual at high risk of cancer. So, it is helping in various diagnoses of bacterial and viral infections, then diagnosis of genetic disorders, then in medico-legal cases, then prenatal diagnosis, and also in cancer. So, diagnosis part include bacterial and viral, genetic disorder, prenatal and cancer. So, all these diagnoses can be done with PCR and also medico-legal cases. So, that is with the application of PCR. Okay, next question, that is DNA fingerprinting. So, you can give the definition and what is the principle and also the application. DNA fingerprinting, the name used for the unambiguous identification technique that takes advantage of differences in DNA sequence. The process of DNA fingerprinting begins by isolating DNA from blood samples, semen, vaginal fluids, hair roots, skin, skeletal remains, etc. And the principle behind it is the variable number tandem repeats. DNA profile uses repetitive sequence that are highly variable called as variable number tandem repeats, particularly short tandem repeats. VNTR loci are very similar between closely related human beings, but it will be different. The analysis of variable number of tandem repeats to detect the degree of relatedness to another sequence of oligonucleotide, making them ideal for DNA fingerprinting. Its application, DNA fingerprinting is applied to pinpoint the culprits of crime like rape cases and murder. Then for the determination of paternity cases, paternal disputes, then in forensic applications, then in diagnosis and developing cures in inherited disorders, then for personal identification. So, mainly in forensic, paternal dispute, and in crime scenes, and also for diagnosis and development for inherited disorders. Okay. Next technique is gene therapy. Gene therapy, it is intracellular delivery of genes to generate a therapeutic effect by correcting an existing abnormality. So, for that is the definition for the gene therapy, that if you are having a existing abnormality, we are delivering a gene to generate a therapy. In order to cure the abnormality, we are delivering a genes to generate a therapeutic effect. Gene therapy replaces a mutated gene with a healthy one. It deactivates a gene that isn't functioning properly. It introduces a new gene into the body to fight against the disease. It enhances the effect of normally functioning gene. Gene therapy activates the gene that was shut down during the fetal lives. So, these are the impacts of gene therapy. Then you can give a brief description on the procedure. Just the summary would be enough, that how genes are being introduced, various method by which the genes are being introduced. First, we are isolating the healthy gene along with the sequence controlling its expression. Then it is incorporated Next, we incorporate this gene into a carrier or a vector. Then it is delivered into the deliver the vector into the target cell. So first is isolation, then it is incorporated, the gene is incorporated into a vector or a carrier. Next it is delivered into the target cell. This vector is delivered into a target cell. So that is the summary of the procedure and how it is being introduced. Various methods. 3 ways are there, that is ex vitro, in situ and in vivo. In ex vitro, where the patient cells are cultured in the laboratory, the new genes are infused into the cell and modified cells are administered back to the patient. In situ, when the expression cascade is injected to the patient either intravenously or directly to the tissue, that is in situ. Then in vivo, the vectors are administered directly to the cell. For example, in case of cystic fibrosis, it is directly administered into the cell. In cystic fibrosis, the gene is directly applied into the respiratory tract cells. Gene therapy was successful for the diseases such as severe combined immunodeficiency, then cystic fibrosis, then hemophilia, cancer, etc. Advantages of gene therapy: the virus is replication deficient, so it is safe and suitable for the treatment of various diseases since the virus is replication deficient. Then disadvantages: there is random insertion can harm the normal cells. Then the retrovirus uses rapidly dividing cells as target. The non-dividing cells are not used. Those are the advantages. Disadvantages. Next technique, that is Western blotting. This can be asked for 3 or 4 marks question. So Western blotting, you need to give the definition first. Western blotting is an immunoblotting techniques which rely on the specificity of binding between protein of interest and a probe which is antibody raised against that particular protein, and this is done to allow detection of the protein of interest in a mixture of many other similar molecules. Steps in Western blotting: first, a protein sample is subjected to electrophoresis on an SDS polyacrylamide gel. Then in the next step, it is electroblotted transfer. This electroblotting transfers the separated proteins from the gel to the surface of the nitrocellulose membrane. In the third step, the blot is incubated with a genetic protein such as milk proteins or BSA, which binds to any remaining sticky places on the nitrocellulose. So first we are doing electrophoresis in order to separate the protein samples. Then it is electroblotted, that is transferring the separated proteins to the from the gel to the nitrocellulose membrane. The next step, we are incubating with the generic protein. The blotted one, the nitrocellulose membrane, it is incubated with a generic protein so that it will bind to any remaining sticky places on the nitrocellulose. Then in the fourth step, an antibody that is specific for the protein of interest is added to the nitrocellulose sheet and it will react with the antigen. Antigen antibody reaction will take place when we are adding antibody for that particular protein. Then only the band containing the protein of interest binds to that antibody and it is forming a layer of antibody molecule. Next is washing up of the excess one. After washing after washing for the removal of non-specifically bound antibody 1, a second antibody is added. Second antibody which specifically recognizes the Fc domain of the primary antibody and binds to it. This antibody 2 is radioactively labeled or covalently linked to a reporter enzyme which allows the which it is allowing the visualization of protein antibody 1 antibody 2 complex. So, after washing the excess which is not bound antibody which is not bound to the antigen, we are adding a second antibody which is radioactively labeled or covalently linked. So, that antibody 2 is being added, so that it is getting attached to the Fc domain of the antibody 1. Primary antibody is getting attached to the second antibody, and thus it is forming a protein antibody 1 antibody 2 complex. And this antibody 2, since it is radioactively labeled, the amount of radioactivity can be calculated, which in turn gives the amount of antibody 1, that in turn gives the identification of the protein. Next is the application. This blotting technique is used in the applications for like HIV test, confirmatory HIV test. Then this Western blot is also used as a definitive test for bovine spongiform encephalopathy. Then in some forms of Lyme diseases is also using Western blotting techniques. Along with the Western blot techniques, other techniques like northern and southern blotting is also important. So, whenever a technique is asked, its application should also be mentioned. Okay, thank you.